at. There we go. Didn't want to record my little story. Oh, it's sticking again. There it goes. Just for a second. For the student view, what you'll be able to see, um, it'll end right here. I have modules for the 16-week class, and they're almost empty because I have scavenged from them. But at the bottom of the 8th week, the 8th module, here's the 3 remaining tests for tomorrow and the next day. Lecture test 3 is the first one. That's tomorrow. That's all text-based. That's at 1 o'clock. Okay. Now, you can click on it all you want. It's not going to let you in. It would let me in, but I don't want to go there right now. 1 o'clock, and you have the exorbitant amount of time. You have the 80 minutes. That'll put you finishing, if you take the whole time, at 2:20. Okay. I have a Zoom meeting scheduled tomorrow at 2:30, 10 minutes after. So you really want to start at 1 o'clock. Okay. If you start a little bit later than 1 o'clock, it's going to let you, but you may not finish in time to come to the start of the 2:30 meeting. If that happens and you have gone over time, well, it doesn't matter. Come back to the Confer Zoom page and select the second meeting. It'll be down here, probably. And then you can join that one. Now, what we're going to do there is review for the practicum, and it'll be probably all images, all of the different types of tissues. Not that we can't do and won't do that today. Okay. That'll be our last meeting. You may think, well, you've had enough reviewing. There's never enough reviewing. If you stay half an hour, okay, that's all, tomorrow, then you'll get credit for the Zoom meeting. Yeah, I know it's only 1 point, but I've had people come at me and say, I'm only 1 point shy. I only needed one more point. Could have gotten it for free. Okay, so stay half an hour or stay all night. I don't care. We can go on until you're exhausted. Okay. And then you can bow out, and I'll get from the event attendance who was there and how long. Okay, we start here on respiratory system. I realize, I think, and hope that that's an easy topic for you. Uh, but give me some thoughts as to what would be best for you in the time we have right now. one thing I can do is to start going through the lecture material and do it fast as possible, actually, or as practical. Why the lecture material? Because it's a lecture test tomorrow. And then I could do more of the tissues later today, or I could do them tomorrow, as you wish. That's one thought. What thoughts do you have? Why do you want to be here except to get 1 point? Why do you, if you want to be here to try to better your score in a real review, what would you like me to review? We've just got so much stuff.
Charlie, I think it'd be great to do a fast review or overview of the lecture material ahead of tomorrow's test.
I agree.
I agree
Yeah,
too.
I agree. Like a whole overview.
Okay,
From lecture 1.
not from lecture
I mean,
1. Lecture
not lecture
1.
1, but for the lecture test.
I
The
got.
last lecture.
Yeah.
Okay, well, here we go. Well, the last lecture I went over yesterday, but we can even do that again if you need it. I don't care. Okay, so actually, I think the first one is the respiratory system, and then we'll have the integumentary system. Okay, and not going to have fetal circulation again. Okay, so here's the first part. Now, I do want to go over things fast. If you need to comment on something, you need to butt in, and that's absolutely fine. Functions of the respiratory system: gas exchange, oxygen and CO2, elimination of waste products, the smell of garlic and ketones, and anything else that contributes to bad breath can come from the digestive system, but it can also come from the respiratory system. To inhale and exhale, the other function of the respiratory system. Respiration, we have two types, external and internal. External respiration is between the body and the environment, and that takes place at the alveolus. That takes place inside the body itself. Yet one portion of it, the air that is inside of the bronchioles and smaller chambers, has not yet entered into the body. It's external. External respiration. Okay, this takes place at the alveolus. Commonly missed question, but once you get it down, you've got it for good. Internal respiration is from the lungs. The blood takes the gases to all of the parts of the body. And here we're especially talking about oxygen. Okay. We could also be talking about anesthetics. Okay. If you've ever had gaseous anesthetics or been at the dentist and had laughing gas. I'm afraid of anesthetics myself. I try not to have them. I won't let them give them to me when I go to the dentist. Not necessary. Just get it over with. But I don't like anesthesia because I don't like the feeling when you wake up from most of them. Groggy, can't think straight. It's scary to me. It's absolutely frightening. So I try to, yes.
You don't get localized anesthetics at the dentist office even?
Local anesthesia, oh, for teeth?
Yes.
Not for fillings. When I was a kid, my mother would drag us down Slauson Avenue in Los Angeles to a dentist that did not use Novocaine on children. This big guy, big stinky guy, frankly, he grabs you, holds you around with one arm, pulls your chin open with the hand of that arm, if you can imagine it, and then puts the drill in. And you drilled with no anesthetic whatsoever. And so I got used to that. I don't like it, but and if I do get anesthetic, it'll be for maybe a root canal or something like that. Okay, but I don't like waking up and feeling crazy and not knowing where I am. I had a spinal last December, and I was sort of leery of that. I told the anesthesiologist I want to be awake during the surgery. This is for the knee replacement. And I want to be awake. I want to see it anyway. You know, it's interesting to me. They put me out. They put me out, and I had no say over it. And when I woke up, it was like somebody turned the television on. It just click, you're back. You know, no, none of that grogginess or anything. So I don't mind that anymore. Okay, internal respiration takes place at the level of the tissues. Well, everything's a tissue. So we're talking about the most internal tissues of the body, not the ones at the lung. That's external respiration. Components of the respiratory system are the nasal cavity, nasal pharynx, oropharynx, laryngeal-pharynx, all three of which are called the pharynx, the larynx itself, and the thyroid cartilage and the trachea with its C rings. Other components, we have, of course, two lungs. Remember that there are three on the right side, two on the left side. The three lobes and two lobes are separated by fissures. Both sides have an oblique fissure. It's the lower one on the right side. And a horizontal fissure is found on the right side, separating the right superior and the intermediate lobes of the right lung. There is no intermediate lobe for the left lung. Approaching the lungs, the trachea divides into two parts, which are called the primary bronchi. Okay. I won't use the abbreviation Roman numeral 1 and a degree sign. It's legitimate to do so, but I'm not going to. I'm going to write it out just like this. You have two branches from the one trachea, and each branch or primary bronchus goes to one lung. Okay. But these primary bronchus subdivide, or primary bronchi subdivide. Three subdivisions on the right going to the three lobes on the right, and two going to the two lobes on the left. They're called secondary bronchi, but you need to know the other term, lobar bronchi, meaning that they go to individual lobes. How many secondary bronchi are there?
5.
5. That's exactly right, because there's 5 lobes. Now, within each lobe, the secondary bronchi divides up into tertiary bronchi. It's just the next level. But there are so many of these, they go in all directions, almost like a spider web. Okay, and as we proceed from trachea to primary to secondary to tertiary bronchi, the tubules are getting smaller. That means that there's greater surface area inside of them and the area for exchange of gas and nutrients. Okay. Rather than being in a big tube like the trachea, in a small one, it's much more intimate. Okay. The primary, secondary, tertiary bronchi have cartilage support to keep them open, as does the trachea itself. So until you get to the tertiary bronchi, which are separate right here, you have cartilage support. After that, you're not going to have cartilage support. the pressure of air coming in and going out will open and close the vessels. After the tertiary bronchi and leaving cartilage support behind, we subdivide many times into the bronchioles, the next smallest passageway, and then to the next smallest passageways, terminal bronchioles. Terminal bronchioles is a misleading name because it's not the end of things. You next have the respiratory bronchioles, the alveolar sacs, and then the smallest chamber is the alveolus itself. The alveolus is the site of the external respiration. You got to remember that one. And sort of a picture here. 1, 2, 3 lobes on the right, 2 lobes on the left, oblique fissure, horizontal fissure here. The names, I think, are pretty self-explanatory for the lobes. The primary bronchus is here, and you have one here that's not being shown by the diagram. Here's the trachea itself. Sort of a blow up, the trachea, the primary bronchus, the secondary bronchus going to this lobe, and then a tertiary bronchus, bronchiole, terminal bronchiole. Okay. Now, this makes it look sort of neat in that you have a single passageway here. That's not realistic. Okay, you have at the secondary bronchiole or the lobar bronchiole, which is here and here, these two. You have many branches coming off. And where the tertiary bronchioles begin coming off, there's many, many of them. And the lungs are just permeated with these passageways. The alveolus is the smallest chamber, represented here as a bump on these blackberries. The alveolar sac would be the entire structure. A terminal bronchiole would be here. Alveolar sac, and then the alveoli. This is showing another picture. Pretty picture. I guess that's why.
Charlie,
Go ahead.
where would the respiratory bronchioles be? I know they're after the terminal, but are they're just not shown?
They're not shown. They're inside.
Gotcha.
Okay. Just know the order. Know the order, and know what's at the end, the alveolus. Now, one thing about the alveolus is that it's the site of gas exchange, sure, but you're only going to get gas exchange with capillaries, and the capillaries do run through here. Here they're showing capillaries running through, okay? But you can't have a thick walled structure. You have to have a very thin walled structure. And in the tissue slides that we've seen, the thin wall of the lung alveolus really shows up. Okay. There's nothing new about this picture. It does look neat, but there's nothing new about it.
So the alveolus is simple squamous as well?
Yes, yes. That's where I was going, and I didn't finish it off. Thank you. You know, you can read many books, and think that, well, it's a book, it's got to be right. But a book doesn't have to be right, not all the time. I proofread a book in plant physiology for my major instructor. He had a very successful book, and it had a lot of experiments. I taught his lab. I ran the experiments. I made the chemicals. I did everything. There were so many mistakes in that book. I don't know how any other school could use it. So I corrected all of the mistakes, gave it back to him, and I was being paid, big deal, $100 by the publisher to proofread the book. I sent back, unfortunately, every correction needing to be made. And when the publisher saw what has to happen, they declined to put out a second edition. I was not very popular. Well, hey, somebody made a mistake, and here's a mistake on this page. You notice the bronchiole looks like it has cartilage. It does not. Okay. Cartilage ends at the stage just ahead of this, the tertiary bronchi. Probably it might have been better for me not to point that out to you, but it would be if you got to studying late tonight and you caught that. Okay. Okay, that was respiratory system. Let's just go down the list. Some of this, of course, overlapped the previous material. The integumentary system is next. This is the skin and the appendages, which are the hairs and the nails. Skin, largest organ of the body. Please remember, I know it seems so easy. The skin is an organ. A lot of people mistakenly call it a tissue and get marked wrong every single time. I'm going to take these 3 tests ahead of you very slowly this time, or slower, and look to see that I get 100% on them. Where I might miss one, I will check it out and see if I made a mistake, or if the question needs fixing. So I'm hoping that we have a set of 3 tests that don't need any discussion when we're finished. Okay, I think that's best for all of us, just to get and see our scores. Okay, skin and appendages make up the integumentary system. We focus on the skin, however, and know that its functions are the protection from abrasion and infection, from desiccation, very important, regulation of the temperature of the body through sweating, and the production of vitamin D. This is the largest organ of the body. The epidermis is the outermost layer, what you would actually touch, and it's divided into 5 regions. And of those 5 regions, you will only touch the outermost one, unless, of course, you've got a cut. This is considered to be stratified because of the number of layers, and squamous because the outermost layers, the one that's flaked off, is squamous in uh, outline. The stratum corneum is the uppermost layer, very thick, 20 to 30 layers. 20 to 30 layers, I say very thick, and I'm comparing one layer to the next. But you know darn well that you can go through 20 to 30 layers of cells to get down to blood with very little effort. The stratum corneum consists only of dead cells, most of them flattened, and they protect against desiccation and infection. The chemical within them is keratin, which reduces the loss of water. That keratin comes from someplace. It comes from layers below. All those layers are coming up, like bubbles coming up from the bottom of a soda can. Okay, everything eventually makes it to the surface, and at the surface, the stratum corneum is formed and contains keratin. Below that, on their way up, the cells are in the stratum lucidum. This can be just a single layer of cells, or it can thicken into up to 40 or even more cells if the skin is abraded a lot, if you do a lot of work, if you form a callus. If you're forming a callus, it's the stratum lucidum that thickens. It also contains elidin. Now, the elidin is a chemical. I have no idea myself what the chemical formula for elidin is, but it's on its way to becoming keratin. And what's going to make it happen when these cells are dead? The cells of stratum lucidum are dead. Okay, what's going to make it happen that there's a change from one chemical to the other is ultraviolet light. Okay, so the closer you get to the surface, the more UV light that you're going to get. How can this layer develop in thickness if you have a callus? The cells accumulate at this level, okay, rather than making it to the surface as fast. The layer below is the stratum granulosum, and it contains the chemical hyalokeratin, also called keratohyalin. I don't know which one I use in the test. I'm sure I do use one. The layer below that is the stratum spinosum. This is a 20 or so layer of cells. They're alive, but they seldom divide. You might as well say they don't divide. I'm not going to put you on the line on this. They may divide, they don't divide. I'm not going to ask you. But this layer of cells, called spinosum, because spine-like crystals seem to pierce from one cell into the next and hold it together like a bunch of tinker toys, if you ever played with those. The only truly living and dividing layer is the bottom layer, the stratum basal. It's the only one of the two layers. Okay. And that divides, only one of the 5 layers that divide. Sorry, I got distracted by the sound. And this is what in plant world we would call a meristem. Most animals don't have meristems. Meristems are areas of continuous cell division. Plants have them all the time. They keep growing their entire life, unless they're annuals. The entire life is just a short period. Okay. You do not have to know the word meristem. Because these cells have the ability to divide, they also have earned the nickname stratum germinativum. Germ meaning the start of something. The entire epidermis is avascular. There's no blood supply up there. That's why I said you have to cut deeper into the dermis before you draw blood. Here are the layers. Let's start from the bottom this time. Here's the stratum basal. Officially, it's considered to be just one layer. But if you have one layer that divides, the next layer is right on top of it, and hard to tell the difference. So keep it just in mind that it's right at the bottom, and it's alive, and it's dividing, and it's forming more cells in the stratum spinosum. The stratum spinosum, which essentially does not divide, okay, well, that means that there cannot be any more cells except those produced by the stratum basal. And the stratum spinosum cells are moved up to the granulosum, from there to the lucidum, and from there to the stratum corneum. The only way any of these layers can get thicker is for them to stop movement of cells through them, but accumulate cells instead. The deeper layer underneath is called the dermis. It's dense, irregular connective tissue. You're going to get asked this 2 or 3 times between now and Friday. Dense, irregular connective tissue. It is vascularized. It contains the sweat glands. It contains the oil glands, or the sebaceous glands. The sweat glands are known as sudoriferous glands. They simply enough provide sweat for cooling off. You may lose some valuable salts or minerals, and so people who do a lot of athletic type work try to replace this or replace the electrolytes. Here's our diagram. Here's the sweat gland. It's a merocrine gland because it produces only a fluid, leaving here by a duct. So it's an exocrine gland. Here's the oil gland. It secretes sebum. It's not proper to call it oil. Everybody does, but not on a test. Okay. Here's the sebaceous gland over on this side. It secretes whole cells in its exudate. Exudate is the secretion. And since it secretes whole cells, it's known as a holocrine gland. It too is an exocrine gland because it has ducts, but the type of exocrine gland is holocrine, merocrine here. The third type of gland is the apocrine, where fractions or pieces of cells are emitted. And the only example we have of that is the mammary gland, which has nothing to do with the integumentary system.
I have a random question. People that sweat a lot, does that mean they have like larger sweat glands? Is that what it is? Or is it just something completely different?
Where's that?
This is just a random question.
Yeah.
I'm just asking, there's individuals that sweat a lot. Does that mean they have like larger sweat glands? Or is that completely different or has nothing to do with it?
No, they don't have different types of sweat glands. I have not told you this, though. There are sweat glands that are larger, okay? It's possible they have more of those, but those sweat glands are located only at the armpit and around the genital area. Those are called apocrine sweat glands. And I did tell you the mammary gland was the best example for apocrine. because that's the easiest one for me to get across. But the largest sweat glands are also apocrine. I don't want to throw in new stuff too much here and go into detail. At this stage, we need to solidify what we've got. Okay. Now, as far as people who sweat more, we used to just call them sweat hogs. Sorry, but
Charlie,
yeah,
I'm just curious. So if someone has like a third-degree like patch of skin, like sorry, third-degree burn, like a patch on their arm or something, does the sweat gland there, does it just stop producing, or what happens?
it will. It will. The degree of the burn is to what depth it goes to. The superficial burn that never gets past the epidermis is just a first degree burn. You get into the dermis, you got a second degree burn. You get into the subcutaneous tissue below that, you have a third degree burn. There's a fourth degree burn. Have you ever heard of it?
No.
It's called carbonization. You don't want one of those. Nothing's coming back. Of course, the trouble with burns is it strips away your protection against infection. You may need skin grafts, but sometimes people who are severely burned, they'll succumb to infection quickly. You know, you would think it'd be very, very painful. I'm sure any burn that you and I remember was always painful. But many of the people who are suffering terrible burns, they have lost all sensation because the receptor cells for the heat and pain are burned away. But that leaves them wide open for infection. These three are showing the types of glands. The Merocrine gland, the sweat gland, produces fluid. The apocrine gland produces partial cells, breaks pieces off. And the production of whole cells in the fluid leaving is the realm of the holocrine gland. When we look at the model of the skin, I believe it shows the apocrine gland, the larger one. And at that time, I'll show it to you. Okay? It is not on the test tomorrow at all. In fact, it's not on the test anywhere, none of the three. But I'll take it in mind to show you that and talk to you about it. Okay, did tissues next? Okay, the next topic we did was nervous tissue. The first portion of this is easy, then it gets a little bit tougher. I'm going to do the first one, and then we'll take a break and continue. Okay, I'm going to do nervous tissue first lecture. Okay, the functions of the nervous system should be, old stuff to you. Conduct sensory stimuli inward, conduct motor impulses away from the CNS, and maintain homeostasis of the body. The components are the central nervous system and the peripheral nervous systems. The central is completely within the dorsal body cavity and consists of the brain and spinal cord. The peripheral nervous system is located in the ventral body cavity for the most part. We do have a questionable area, and that's where the preganglionic neuron emanate from the lateral intermediate mass, okay, inside of the spinal cord. Okay, the two are lying side by side. Okay, so where do we say the peripheral nervous system is? Well, the textbook answer is that it is in the ventral body cavity. Okay, I have not asked that question of you because I think it can be debated. The peripheral nervous system has two parts. At first, the afferent nervous system. This is incoming of information. This is sensory information. Okay. This could be coming from your arms, your legs. It can also be coming from inside your body, from the organs inside your body, telling the brain and spinal cord how they're doing. The efferent nervous system has motor neurons. So it would come out the ventral root, if you recall that. Okay. This includes the autonomic nervous system as well, which innervates smooth and cardiac muscle and glandular epithelium. Please remember these three guys. And we know that the autonomic nervous system is divided into two parts, sympathetic and parasympathetic. Okay. Also part of the efferent nervous system might be the cranial nerves. It can also be part of the afferent nervous system, and I should have put it up here because some of the cranial nerves go in both directions. Okay. You're not going to miss any questions because I didn't put it up there. Trust me. Here's the organization then, and it's just a schematic of what I just said. The brain and spinal cord are part of the CNS. Everything else is part of the peripheral nervous system, including the afferent fibers going in and the efferent fibers coming out. Those efferent or motor fibers, some of them go to the muscles. This chart calls it the somatic nervous system. I think it's more realistic to call it the voluntary nervous system. And the efferent fibers also split and go to the autonomic nervous system. That's sometimes called the involuntary nervous system, but autonomic rubs everybody better. The autonomic is broken into the sympathetic or thoracolumbar system and the parasympathetic cranial sacral. Glial cells. They're also called neuroglia. 90% approximately of the brain cells are not neurons, they are glial cells. And they have the functions that we talked about just a moment ago, which is nice. The astrocytes, that's this one here, with arms extending in all directions, can hold something in its right hand, something in its left hand, something in its backhand, something in its forehand, and really acts as the cement inside of the central nervous system. These glial cells are only in the central nervous system, so you can think brain and spinal cord here. The oligodendrocytes are also only in the central nervous system, and they form the myelin sheath. By that I mean here with the blue, they have extensions that wrap around the axon of, in this case, a multipolar neuron. The axon, if it's myelinated, would not be myelinated and then there's a gap and then it's myelinated again. It would be myelinated its entire length, so we're missing one. right here. They wanted to show us the axon. This here is an astrocyte. So the oligodendrocyte that makes the myelin sheath does not have a lot of extra dendrites or arms to attach to things. So it would not be very good at holding everything together. So along with the oligodendrocytes, we need the astrocytes anyway. The microglial cell is the small one here. It's does have the ability to bind things together, but its primary chore is to phagocytose and get rid of foreign objects or objects that do not belong any longer in the central nervous system. Red blood cells that have broken down, bacteria that may be there, anything else that is not useful, this will take care of it. These are capable of diapedesis. The types of neurons, the first one is the multipolar neuron. This particular multipolar neuron is in the peripheral nervous system. How do I know that? Because the myelin sheath is made up of Schwann cells. It says so. It's going to have the same function, but its function is going to be limited in its location to the periphery. Here's a dendrite bringing information in. Here's the cell body, also known as the parakaryon. Don't worry about a mitochondria. Every cell has to have those. And it's got a nucleus too. The axon hillock here is where the depolarization that the dendrite experiences accumulates. We need depolarization of probably more than one dendrite in a multipolar neuron, of several. And the depolarization accumulates here. And if there's enough, it shoots off an impulse along the axon, also known as the axon cylinder, until it reaches the end where it can branch out into axon branches. They're using the term axon terminal. The best term is actually teleodendria. Teleodendrion is singular. They have the term synapse here, and there would be a synapse at the end of every teleodendrion. But they haven't drawn it. They're showing you where it is, but the picture does not show the synapse. A bipolar neuron has only one way in and one way out. Here's the dendrite that receives the sensory information. And if it receives enough sensory information, it sends it along to the cell body. There's no axon hillock this time. It passes right through the cell body and down the new axon to the axon branches or teleodendria. There is no modification, no chance for modification. The sensation that is delivered to the cell body is the same sensation that leaves it. So these might be found in the eye and in the ear, where pitch stays the same, where red stays red. Yes, I know we get multiple colors, we get different shades of colors and things, but we don't have as many bipolar neuron types as we do colors. So it's our mind itself, once it receives the information, that will become our palette and blend things. the pseudo unipolar neuron. Okay. Has incoming dendrites.
Oh, Charlie?
Yep.
I actually have a question on the image before this one. It looks like when it's pointing to the, well, where it says dendrite, it looks like it's pointing to the, oh, no, sorry. You just passed it.
That's
Yeah,
me.
it was that one. Yeah. That one right there. So where it says dendrite, it looks like it's pointing to the axon, or is that because it's just prior to the cell body that is dendrite?
Okay. I think you just caught one of the things I was telling you. You can't trust every book, and I love the picture. It gets the idea across. One type of input, same type of output. That's what I wanted to get across. But this really, to me, is an axon. Okay, they can go in both directions. Okay, and this really is an axon, and it's moving right on through to the other end. Where's the dendrite? The frilly stuff. Okay, can you handle that?
Yeah.
Okay, but I will not be asking any questions that put you on the line with that. Okay, so in this case, this pseudo unipolar neuron, where might we find it? Well, from the fingertip that touches a tack, okay, and has pain. The pain will come down, may even speed up here with the Schwann cells encircling this axon. This is an axon also. This is the dendrite here. I'm not going to ask you about the precentral axon, the one before we get to the middle. Okay. But here's the Schwann cell. And then here's the cell body, swollen, bigger. And when we have a true, not neuron, but nerve, we're going to have hundreds and thousands of these cell bodies all at the same point. And they're going to balloon the area. And that's what forms the dorsal root ganglion. But the impulse continues on. And through the myelin sheath, through the true axon, they call it, to the axon terminal, which I prefer, again, teleodendrion. I'm looking at these to find nerve, afferent fibers, where they go, efferent, somatic, visceral. I think we don't need to stop on these guys. Here they're using a new term, peripheral process and central process. You can just call that whole thing axon. You can call it that, but I'm not going to hear it because it's not on the chest. Please know what an effector is. A lot of people get that confused. An effector is something that is effected, not something that does the change. Synapses, these occur between neurons from the dendrion to the next dendrite. So here's the cell body. Dendrite's not drawn. Axon, axon branches, space is a synapse. We have a better picture coming up. Gray matter, okay, the part of the central and peripheral nervous system that is not myelinated, it moves more slowly. White matter has myelination. Impulse moves fast. Ganglion first, collection of nerve cell bodies outside the CNS, or you can say within the PNS. But if you have a collection of nerve cell bodies within the brain or spinal cord, it's a nucleus. Well, that was the end of that. Okay. I'm going to go through the rest of it. I don't know what we need to do from yesterday, but I'm going to. And I'll start that, so 28, I'll start that at 20 of 3. Now, as far as your credit is concerned, you have it already. Okay. If this is valuable to you, please come back. If it's not, you're welcome to go and do whatever you're going to do, study, not study, work on your study guides or outlines and get those in. Okay, but at this point, everybody here has will be given the Zoom credit. Okay, and I'll see you in about 10 minutes. Okay.
Hi, Charlie.
Howdy. Okay. Back to the marathon, unless you have a question. Okay. If you do, just shout it out anytime. Okay, that was the nervous tissue beginning lecture. Then we did the autonomic nervous system. Okay, or the nervous system part 2. Chapter 12 helps you out with this one. If you haven't yet done chapter 12 or done the outline, you're thinking, I don't need extra credit. I'll just take these tests on my nerve. That's fine, but chapter 12 will still help you with this. The autonomic nervous system, okay? First, it starts by referring to the afferent nervous system. That's not autonomic. The afferent nervous system is the incoming one. Sensory stimuli are brought into the CNS. They cross two neurons only. I'm sure there's some that have more, but for the most part, two neurons to get from uh, touching something hot to getting it into the central nervous system. When something gets into the central nervous system, like the brain, it may spin around from neuron to neuron to neuron and use what we call interneurons or in between neurons while the brain mulls over something. I don't think a brain mulls over a burn for very long. But if you've ever played chess, it's like it won't stop. It keeps thinking and thinking and thinking. Okay. The neurons that are coming in from the afferent nervous system are really coming in by nerves. Okay, nerve is a collection of neurons. If it's coming inward, it's afferent or sensory. Okay, if it's going in the opposite direction, outward, to innervate either skeletal muscle, smooth muscle, or glandular epithelium, then it's an efferent nervous system, but it's a nerve also. We can call it a motor nerve. Okay. Having a single neuron synapsing with the next single neuron is not going to work. It's going to break sooner or later, and then you'll lose that ability to sense things. That's why we have redundancy, plenty of redundancy in the nervous system. You know, that sympathetic nervous system we talked about for days now, being able to move up and down the sympathetic trunk ganglia, or the chain ganglia as they're also called. They can move up and down, that's true, but they can take a sensation that was initiated by the brain and scatter it throughout the rest of the nervous system. And if you do have an injury at one point and break an entire nerve, doesn't mean that you will lose the ability to function uh, um, at that point, because the function just be translated up or transferred up to the next level or down a level. We have these areas in the body called plexi. Plexus is singular. And these are like switching stations where we can take the information that's coming in and send it to many places. Okay. Motor commands reach the involuntary nervous system as well as the voluntary. If they reach the voluntary nervous system, they'll have an effect or innervate skeletal muscle. If efferent nerves reach the autonomic nervous system, they innervate smooth muscle, cardiac muscle, or the glandular epithelium. This little chart, I like the other one better actually. This is more colorful, but I like the other one better. Here's the central nervous system over here. This is the peripheral nervous system here. You know, they could really take this out and shift this whole thing in the two arrows there, one in and one out, right to there. And it would make just as much sense. But the peripheral nervous system, yes, does go into, by the blue line, the CNS. Then out comes the motor impulse. Motor impulse or efferent division is divided into the somatic nervous system. We call that the voluntary nervous system. And the ANS, the autonomic nervous system, which subdivides, of course, to sympathetic and parasympathetic. The incoming or afferent nervous system consists of a somatic sensory. Okay, so sensations from the body are also sensations from the organs, visceral sensory. And then we have totally the sensory division or afferent division, which takes the information to the brain. Again, I really think this little box is probably unnecessary. This is showing you a 2-neuron run, okay, 2-neuron run, and these 2 neurons are going to be in the autonomic nervous system. Okay. This first neuron, which here, this part is buried in the spinal cord at the lateral intermediate mass. We'll see a better shot of that in a moment. This is the preganglionic neuron. Can also be called the presynaptic neuron. But since we know that neurons don't just run by themselves, there are many, many of them, then yes, we can point to a synapse. But what we're really going to have here is a ganglion, a group of neuron pairs that are synapsing. There's nothing wrong with this diagram. It's just we've got to be sure that we understand it. Here's the impulse or action potential. Splits up at an axon branch, crosses a synapse, and then it goes on to the postganglionic neuron. That is being called here postsynaptic cell. It's the same thing, exactly the same thing. At the end of an autonomic neuron pair, there will be a myo-neural junction. The neural part is right here. The myo part is the smooth muscle of the organs, such as the stomach. If I do this in a little bit different order this time, the neurotransmitter at the junction between the pre and post ganglionic neuron is always acetylcholine, whether we're talking about the sympathetic or parasympathetic system. It's always acetylcholine. Get down here to the myoneural junction though, and the neurotransmitter is still acetylcholine, still the slow one in the parasympathetic system, and it's noradrenaline in the sympathetic system, and it has much more pronounced and faster effect. Here's the synapse. Here's the end of an axon branch or a teleodendrion. It has vesicles inside that are filled, primed with neurotransmitter. when this is stimulated, the neurotransmitter is released into the gap. It's called a synaptic cleft. I never gave you the term, so you know it's not going to be on any test. And then it moves across. This is really just by diffusion. It has to move across this gap. Diffusion is a slow process, and so crossing this gap slows things down. That's why you don't want to have too many of these synapses. Unless you're in the central nervous system and you're thinking. That triggers a response or a depolarization on the other side in the next dendrite. This one, spinal cord, really only shows the dorsal root ganglion and the dorsal root. They're incoming. Ventral root is outgoing, and where the two come together and run as a mixed nerve, that's the spinal nerve. Most nerves are mixed nerves. The other thing's not labeled here, but we do have it in the next lecture, is this bulge inward here on the horns. This is the lateral intermediate mass. And the preganglionic neuron originates there. It's not doing that here, is it? It's coming in from the afferent or sensory and going straight across. Well, this is a reflex arc. This has nothing to do with the autonomic nervous system. It's not using that. It's immediately using an interneuron to shuttle across and get a reflex to save something like a burned finger. Okay, this tells us nothing that we shouldn't already know. The sympathetic nervous system is the thoracolumbar, parasympathetic, craniosacral. This is a comparison, first in names, thoracolumbar versus craniosacral for the parasympathetic system. The sympathetic system excites and prepares the body for fight or flight, but the parasympathetic system dominates in less tense situations. Both of these systems work all of the time. It's which one predominates. In the sympathetic system here, the preganglionic neuron, we just saw two. The two we saw a moment ago were about the same length. In the autonomic nervous system, though, that really isn't the case. The preganglionic neuron is short and terminates in the chain ganglia. The postganglionic neuron, on the other hand, is comparatively long. Whereas in the parasympathetic system, the preganglionic neuron is the one that's long. And it may terminate in a ganglia near the organ, right next to it. Since the sympathetic neurons and the myoneurojunction have acetylcholine and then noradrenaline, as we just said, the system is considered adrenergic for adrenaline. In the parasympathetic system, we just said that acetylcholine crosses both the synapse and the myoneurojunction, and it's relatively slow. And so we call this system entirely cholinergic. Okay, so if we look at the sympathetic system first, always the easier one actually, here's the thoraco area. We count them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. That's weird. They should have 2 more, but it doesn't matter. These are the thoracolumbar vertebra, and the preganglionic neuron that comes out of them is relatively short and terminates in a chain ganglion. This is the sympathetic chain, as it's called. Once an impulse sent out, a motor impulse, reaches the chain ganglion, it can go anywhere, all the way up, all the way up, all the way down. 31 pairs of these chain ganglia. Okay. But just take one, and the preganglionic neuron is relatively short, and the postganglionic neuron is long. In the craniosacral system, the parasympathetic, you usually have a long preganglionic neuron followed by a short postganglionic neuron. Long, short, long but not real long, and shorter.
Charlie?
Yeah.
In the somatic system, does the route of the neurons, is it more similar to that of the craniosacral?
I didn't hear the first part of what you said. It didn't come across.
My apologies. In the somatic system, are the preganglionic neurons long and the postganglionic short, or the opposite?
You said in the somatic system.
Correct.
What are you referring to?
Like the voluntary system?
No, has nothing.
Muscles?
That's what I thought. Has absolutely nothing to do with the voluntary system.
Right. I know this doesn't. I'm just asking in regards to the voluntary system. Is the path, is it anywhere near one of these systems or just completely different?
No, it usually has a 2 neuron pathway also for the motor, but the length of the neurons will vary depending upon which organ pathway you're going to. So it's really not comparable.
Okay, thank you.
Okay. Here again, we see the sympathetic system, acetylcholine, and then noradrenaline. Whereas with the sympathetic, parasympathetic system, it's acetylcholine in both cases at the ganglion and also at the myoneural junction. The cranial nerves. I think there are 5 questions, I've even said it here, on the test. That'll be tomorrow's test. 5 of these will be picked, and you'll have to come up from a multiple choice set with the correct name over here. Pretty straightforward.
That's for tomorrow's test?
Yeah. Yeah, everything we've been doing is for tomorrow's test. Everything that we've been doing today is really for tomorrow's test. Okay, now I need to at some point go over histology. That'll be on tomorrow's test too. I'm not talking about the pictures. Okay, I'm talking about what the word simple means, what the word compound means. And so I'm asking you, do you want me to go there, or do you want me to, okay, where is it? Well, I reviewed this yesterday. Where here. The spinal nerves and the ANS. I reviewed it yesterday. Do you need it 2 days in a row?
Yeah, sure. I'm down for that.
Charlie, I prefer to do the histology question since we did just go over that yesterday.
Yeah.
Me too.
Okay. 2 to 1 so far. I'm going to break the tie and tell you, if we don't get to this, I did it yesterday and it's recorded. Okay. It made the recording. Trust me. I sort of cringe at doing it 2 days in a row. It's like, if it doesn't sink in once, I don't know if it'll sink in twice. Okay, so I want to go back up for right now and do the tissues part 1. It says lab. Okay. But it has some lecture material in it. Okay, here's the tissues and organs. First off, don't forget the simplest thing you would think, the 4 types of tissues. It's not the simplest thing. People forget it all the time. You ask a simple question, they think it can't be that easy. And then they go and miss it. So you want epithelial, connective, muscle, and nervous tissue. The epithelial tissues have the most that needs to be said about them. A number of types of these that are named and judged by their shape and their arrangement. The basic shapes are cuboid, columnar, and squamous. Okay. This is the cuboid shape. These do look like little cubes. I always thought that, and I told people, they look like marshmallows. But then somebody told me marshmallows are round, cylindrical. I had to go out and get a bag of marshmallows and see for myself. They are. Okay. So instead, think of this maybe as a baking sheet. You've baked the cake and now you're cutting it into brownies. Okay. And they're all cuboid in shape. That's the first. This is a simple cuboidal epithelium because it has only one layer. If I were to put a second layer on top of this, it would be a compound cuboidal. And if I put more than 2, it's going to be stratified cuboidal. Okay. But a simple cuboidal is what we normally find. Sometimes it lies flat like this, simple cuboidal, but usually simple cuboidal forms a tube and it rolls itself up. Okay, so it looks like this over here. Okay. These are simple cuboidal cells. You can see the cells along the side as well. The lumen is on the inside, but the basement membrane, here called basal lamina, the basement membrane that all epithelial cells have to attach to, is on the outside here. We find these primarily in the kidney tubules. Now, I told you that you're not going to get pictures tomorrow, but you will get these questions that say, where is the simple cuboidal apt to be found? And the answer would be the kidney tubules. Or I could reverse it and say the kidney tubules are formed from what type of epithelium? Simple cuboidal. Okay. These are all simple cuboidal here. This is a nice set, simple cuboidal. Simple squamous would be one cell layer. That's what the simple means. And squamous would mean flat. And it's here at the Bowman's capsule. Very, very thin. One cell layer thick. You see several nuclei. And so Bowman's capsule is simple squamous. Most simple squamous allows for transfer. But here the Bowman's capsule is primarily for containment of the plasma that's being expressed by the glomerulus, which is a capillary network, part of the circulatory system.
Charlie, since the kidney has different parts of it with different parts of epithelial tissue, will you be specific as to which part of the kidney you're asking?
I really don't have to be, because it's all you're going to find in the kidney is simple cuboidal. Let's see, I'm trying to think, where would you find anything else? Oh, on the outside, but we're asking inside the kidney. The
But the
collecting duct.
glomerulus is simple squamous?
Oh yes. You will know what I want. In other words, if I want the Bowman's capsule, the answer's got to be simple squamous. If I want kidney tubules, it's got to be simple cuboid. Okay, I'm not going to let you be guessing. These cells are epithelial cells of the villus or villi. These outer cells are called as a group microvilli. This is really an excellent picture here. You can see the individual cells. They're elongated, they're simple, one layer thick, and they are columnar. As long as you know that this is digestive system and it's not the upper esophagus, which I would tell you, then it's got to be lined by simple columnar epithelium. The entire GI tract is, except the upper esophagus. We're not doing compound cuboidal or columnar. I've told you where it was if you're interested. Stratified squamous. Here's our skin once again. Not just the whole skin though. This is the epidermis. And the outermost layer, the stratum corneum, has the flattened cells. The stratum lucidum is shown here with flattening cells as well. These are stratified, many layers thick, and flat, so they're stratified squamous. Transitional epithelium. This will change its shape or distend, enlarge when it inflates, if I can use that word. You find it in the oviduct, you find it also in the ureter. Here's the tissue. It's multi-layered, so it's stratified. When it's stratified, it can only be one of two things. Stratified squamous, in which case these cells should look like tiles. They should be flat. And if they're not, then it's transitional epithelium. This can change its shape as the epithelium stretches, as the ureter fills with urine and enlarges like a balloon actually. These will change their shape. They will become flatter, but they will come back. Why don't we have flat cells here, fully distended cells here? Because when you take the ureter from an organism that you have sacrificed, it relaxes and comes back to this shape. You're never going to see transitional epithelium that is flat. Pseudostratified ciliated columnar epithelium. Pseudostratified, fine. Ciliated gives it away. Here's the cilia. And here's the first layer. It's one layer thick. I know it looks like more than one. That's why they call it pseudostratified. But with focusing up and down on the microscope, you can trace any object to the basement membrane. They're all attached to the basement membrane, which is right here. So this is pseudostratified ciliated columnar epithelium. And this kind of cell is found in the respiratory system. It's found in the nose, in the nasal cavity. It's also found in the trachea and in the larynx. That was it. From there, we went on to some connective tissues. Now, these pictures you'll want to be familiar with on Friday. I'm sorry, not Friday, Thursday, for the first test at 1 o'clock. But we pass sort of by them right now and get the terminology instead. This is the relar connective tissue, usually found in soft parts of the body that are flexible. Adipose stores fat, works against trauma, insulates the body. Realize, of course, that one person's slide that they made of adipose can look completely different from another. So what's the similarity? The big white area, because you don't have any inclusion except lipid. I was going to say also that after the test tomorrow, that's at 1 o'clock and ends at 2:20, 2:30, I'm going to have a session, as I said, half hour gets you the credit, but that's only for photos, these pictures, okay? And that can go on as long as we need. Here's dense, regular connective tissue. You would find this in the tendon. That's all you need to get out of it for tomorrow. Dense, irregular connective tissue. I think I told you you're going to see that definitely on the test. It's the dermis. The dermis. That's this area down here. Mostly, I guess, pink. It has sweat glands located in it. It has oil glands too if we had hairs, but we don't have oil glands in this slide. This is the bottom of the epidermis right here. Goes all the way up to here. This is the basement membrane in here. I know there will be a question tomorrow. What kind of tissue makes up the dermis? And and the answer multiple choice, dense irregular connective tissue. Okay, here's our compact bone and cancellous bone or spongy bone. Nothing really we need to say there. We talked about hyaline cartilage yesterday. The main thing that's needed for the written test is that it's avascular. Okay. Avascular, and it makes up the protection of the articulations, articular surfaces. It's the most common type of cartilage that we have. The elastic cartilage is found in two places. It's found in the ears, and it's found in the epiglottis. A lot of people, I almost shouldn't say it, would put down nose, but they'd be wrong. Okay, that's hyaline cartilage. Okay.
Charlie, what kind of tissue is the upper half of the esophagus?
Stratified squamous.
Okay.
Okay, make Mike happy. I'm going to do this really quick because I did it yesterday. Okay, spinal nerves is shown in yellow here. There are 31 pairs. You have 8 pairs in the cervical region. You have 12 in the thoracic region, 5 in the lumbar, 5 in the sacral, and 1 more in the coccygeal region. That's 31 pairs. Okay. Okay. I was just taking a moment to mute someone. Okay, the spinal nerves that we see here are all in the peripheral nervous system. Spinal nerves are all mixed nerves, definitely mixed. They're in the peripheral, so incoming and outgoing. Incoming to the central nervous system, outgoing from the central nervous system. The vertebra, we got tested on this before. Just have to know the names of the parts here. This diagram, you definitely need to know the name of all the parts except the one that says not covered, the collateral ganglion. You can do without that one. But you want to expect to see a diagram like this tomorrow and to be able to name it. This is showing the movement of neurons from the sympathetic system. They start at the lateral intermedial mass. The preganglionic neuron is coming out. This is an efferent system. This is not an afferent system. The autonomic nervous system transmits motor impulses from the brain and spinal cord. So it's an efferent system. Goes out through the ventral root, enters the spinal cord. Enters the spinal nerve, comes across, passes by this one, and goes into the next tunnel which is the white ramus, the white arm. There it ends, and it can synapse with the postganglionic neuron, either right there, or it can continue on and go down or go up, and realizing that this is actually a nerve and not just a neuron, you can split both ways. The preganglionic neuron here enters in through a white ramus. If it synapses there, here comes the postganglionic neuron in blue, and it'll immediately go out the gray ramus and continue to the organ of the body that's supposed to be worked upon. This sort of in and out can happen all the way up and down the shade ganglia, because the preganglionic red neuron or nerve can split and go in all different directions. And if it ends up in a different ganglion, it can synapse there, and out the postganglionic neuron will come through the gray ramus. White ramus is in only, the gray ramus is out only. The white ramus is only found in the thoracolumbar vertebra and system. In other words, the sympathetic system is the only one that can initiate a neural motor response that can find a white ramus to get into the trunk. Once in the trunk, it can go all the way to the top of the body or to the tail. There are no white rami entrances, doorways in any of the other vertebrae. None in the sacral area, none in the coccygeal area, and none in the cervical area. They cannot send their impulses into the chain ganglia. So they have no influence up and down the body. They'll have to stay put. Here's that situation here. The preganglionic neuron comes. Here's the sympathetic trunk ganglia. Let's say somewhere down in the sacral area. It's got a gray ramus, but it doesn't have a white ramus to get in. Information passes by and moves by way of what's called a splanchnic nerve out to the organ without changing, without interacting at all. Well, what's this doing down here then? The sympathetic trunk ganglia that might be in the sacral region or the cervical region can receive information from the thoracolumbar region, okay, which can access all of the chain ganglia. And then you can get your synapse all the way from pair 1 to pair 31 of the spinal nerves. And then out that can go to the gray ramus. And now it can act at this level. The sympathetic system can act at every level because it can get on this elevator, as I call it, these chain ganglia. The parasympathetic system can only act at the level that initiates it. It cannot get on to the sympathetic trunk ganglion elevator. Okay, those that are still with me, okay, do you have a preference? I didn't go over the endocrine system. It's simply to learn this chart, name of the endocrine gland, hormone that's produced, target organ, and effect. You do also have to know what a hormone is and the characteristics of a hormone. Actually, that first part's dead easy. It's a little bit tougher getting all these abbreviations and things down. Let me try something different. Okay, I'm going to pick on somebody. Sorry. First on somebody I can see, then on somebody I can't see. Jessica, you there?
Yes.
Okay. What do you intend to do to study tonight, if anything, or watch the Olympics?
I'm gonna watch the video from yesterday, I think it was. And then I have some of the slides printed out. I'm gonna go through those.
Okay. Let's see. Kyle, are you there? Christina. Nick, you're lit up. I don't think anybody's there. If you've got your video off, would you at least, under reactions,
I'm here.
oh, you're always here, Mike. Sorry.
I'm here.
Okay, how are you going to do it? What are you going to study tonight?
I'm gonna be going, or actually finishing up the histology, like packets that I print out, and then afterwards I'm gonna be going over. I think it was like the what was it? Where we went over the histology stuff, like more of the histology stuff.
Okay. You're going to have plenty of time tomorrow night for histology too. So if you find it's taking up all your time and you're not feeling 100% about the lecture test, I'd go with the lecture test. Okay. Casey,
What would you recommend for like even like tomorrow, but like I would say like 3 hours before the test? What would be like a quick review thing that would, almost like all in one, would have the most material that I could easily overview?
well, I would actually tell you something you won't take my advice, and I know you won't. I wouldn't take my own advice when I was in school. I was a crammer. I had to know what to cram. And your question is a question I would ask. But I'd tell you, get it done tonight and relax tomorrow. I'm sorry. I think relaxing is important before the test. So whatever section you feel least competent in, that's what you got to study.
Got it.
Okay. Okay.
Oh, also a question. you know how you have it's like the 50 questions of the practice questions for the final?
Yeah.
Is that more like, because I know all the questions are going to be kind of like universal throughout the 3 tests that we're going to take. But are those 50 questions, are they more focused towards like this first lecture test that's coming up or the final exam?
Neither.
Neither? Okay.
I start the test with maybe, let's say, question number 1. I look at the first term on the list of terms. I design a question. Then I skip 5 or 4, let's say 4. Then I take that term and I make questions. Skip 4 more, 4 more. So I can get all the way through to the end equally. Questions that or topics that get more than one question, they end up having to have been longer topics. Okay, so they're all going to get asked. It's not skewed towards the beginning or the end. It's everywhere.
Got it.
Okay. Allison, what do you have left to do?
What I'm gonna do is I have all of the lecture, I have pretty much every single lecture printed out. So I'm gonna go over the outlines I made for 12 and 15 and just kind of reread the notes as much as possible.
Okay. I think the outlines for 12 and 15 should be good.
Okay.
I would ask some of you, but I know who's already completely studied out. Okay, either ask me something, ask me to do something, ask me to show you something, or I guess we'll go. Kind of awkward just sitting here.
Charlie, this was such a good review. I really feel confident moving in tomorrow because of some of the things you brought up today that reinforced what we had learned previously. So thank you.
Okay, glad that helped.
Thanks,
Charlie.
Okay, either Jessica.
Oh, I was saying bye.
Oh, bye.
Bye.
Okay, Mike.
This is kind of off topic, but I thought it was funny to share. I don't know if I'll remember to tell you tomorrow, but I think it was like on one of the lecture tests, I think I was the only one who was the last one to log out. And that was because I fell asleep, I think 5-10 minutes at the end of the lecture. And so once I woke up, I was like, oh no, I didn't even log off. So I went back to review the video, the recording, and I think it was just you and me in the session for like, I think 3 minutes until you left, and I felt
I
very
gave
bad.
up.
Yeah, sorry about that.
That's okay. I usually give a shout, to who's ever last. Last is usually you or Taryn or Randy or Allison's close to it. Emmy. Those. It doesn't matter who's last. It doesn't matter. It's matters who's first on the exam. Okay.
Well, thank you, Charlie.
Goodbye.
Thank you.
Thank you, Charlie.
Bye.
Charlie.
Thank you.
Thank you, Charlie.
Goodbye.
Thank you.
Do well tomorrow.
Thank you, Charlie. See you tomorrow.
Okay.
Not last today. Bye. Charlie. Thank you.
Okay. Are you there?